This is Dirk Sieger from the University of Edinburgh, and I'm happy to record this webinar for the World Cancer Day. So I'm going to tell you about our research on macrophages and microglia and their role during brain tumor growth. But before we get started, let me briefly introduce macrophages and microglia. So macrophages and microglia are part of our innate immune system. These cells are professional phagocytes, and they provide the first line of defense against any sort of injury or infection in our body. And as you can see here, these cells basically can be found in almost every organ in our body where we have a resident population of these macrophages. And these macrophages are meant to protect our organs, but at the same time, they're involved in homeostasis within these organs. The microglia are the resident macrophages of our brain. And these cells, as you can see here, are highly active. They constantly scan their environment by sending out and retracting their tiny little processes. And apart from the classic immune functions, these cells have been shown to be involved in neurogenesis, for example, and in axonal outgrowth, in synaptic refinement, and in the phagocytosis of apoptotic neurons. So overall, these cells have a lot of beneficial functions in our brain. However, microglia can also play a detrimental role, and that's obvious during tumor growth in the brain. And over the past 10 years, there's been a lot of research looking at macrophages and microglia within glioma, which is one of the most aggressive brain tumors. And basically what researchers could show is that these macrophages and microglia, they infiltrate these brain tumors. And instead of attacking or fighting the tumors, they actively promote growth of these tumors. And over the years, a lot of signals have been identified that mediate this very complex crosstalk between the macrophages and these tumor cells. And the bottom line here is really that macrophages and microglia exhibit a lot of function that promote the growth of tumors. However, most studies have only looked at late-stage tumors that are heavily infiltrated by these microglia and macrophages. So what was missing so far was the early understanding when microglia and macrophages contribute to tumor growth. And if you think about this, in theory, there are two options here. So you either might be looking at a solid tumor, as schematically shown here, and these tumors release a lot of cytokines and chemokines, which lead to the attraction of microglia, to their infiltration into the tumor. There might be additional macrophages coming in later. And once within the tumor microenvironment, these cells develop their pro-tumoral activities. However, that's only one option. And what we were speculating years ago when we started our studies is that, as you have just seen, these microglia are densely covering our brain anyway. So we were speculating that as soon as abnormal cells develop in the brain, these microglia should be able to detect these cells. And there might even be an early infiltration of macrophages. So we set up to understand when do microglia and macrophages respond to tumors in the brain, what are the signals, and what is the nature of this initial interaction. So is it anti-tumoral at the start, or is it immediately pro-tumoral? We've addressed these questions in a zebrafish model, and I'm not going into details here. So basically what we have is we have a transgenic background that allows us to overexpress oncogenes to induce tumors. In this case, we have used the human AKT1 gene with an additional fluorescent tag, so we can visualize these tumor cells. And as you can see here, this works pretty well. So within a month's time, we detect solid tumors in the brains of these fish. But more important for our work is that we can actually detect the first abnormalities immediately after oncogene activation. These images are taken a week after oncogene activation, and you can see that these AKT positive cells, in comparison to control cells, they become abnormal, they are bigger in size, they are irregular in their shape, they have high levels of the human AKT1 protein, and importantly, they increase their proliferation immediately. Meaning this really allowed us to address the earliest responses of microglia to these tumors. We have done that by using a microglia-specific antibody and compared healthy control brains to these early AKT-positive tumor brains. And as you can see here, within a few days upon oncogene activation, we get a significant increase in the microglia population size. And at the same time, we get a massive infiltration of new macrophages into these brains. And to cut a long story short, we have several lines of evidence here showing that this increase in the microglia population is caused by this infiltration of new macrophages. And we can actually show that these infiltrating macrophages then differentiate into microglia or microglia-like cells. Just showing one of the examples here where we have used a double transgene that allows us to label macrophages, including microglia with a red fluorescent protein here based on or under control of the MPEG promoter, combined with a microglia-specific transgene that is only active in the microglia. And indeed, as you see here, we can detect macrophages that are, when they're at the periphery, negative for the microglia marker, but once they infiltrate the brain, they become positive for the microglia marker and differentiate into microglia-like cells. We have identified the underlying mechanism, so we know how these cells are attracted to the brain. We have isolated these AKT positive cells, these early tumor cells from the brains, FAC sorted them, and performed qPCR here. And this allowed us to show that these cells release high amounts of SDF1, and this is the signal which attracts macrophages via the activation of their CXCR4 receptor. And we can prove this by using a zebrafish mutant that is devoid of the CXCR4 receptor. And as you can see here, the mutant itself has a normal number of microglia. However, upon tumor induction, we don't see any increase in the microglia population size here, and we don't see any infiltration of new macrophages. So it's specific SDF signaling from these early tumor cells that recruits the macrophages into these brains. But what are these macrophages and microglia actually doing? Are they anti-tumoral during these early stages? Are they clearing these tumor cells, or are they promoting their growth? And we have addressed this by performing our experiments in a microglia and macrophage null background. We've used the IRF8 mutant here, which is devoid of macrophages and microglia during these developmental stages, as you can see here. And importantly, tumor induction in this background without any macrophages and microglia looks very different. So we have assessed the proliferation of these early tumor cells, of these AKT cells here. And as you can see, we see a significant drop in the proliferation of these cells in this macrophage microglia null background. Meaning, even during these very early days, there's a clear tumor promoting activity coming from these macrophages and microglia. So we know they're promoting the proliferation, but how are they actually promoting proliferation of these tumor cells? And we're addressing these questions now, and for the next couple of minutes, I'm going to show you some newer unpublished data on that. So what we have observed during the time is that the microglia macrophages show long-lasting and direct interactions with these early neoplastic cells. You can see this here. So there's a lot of crosstalk. You see these microglia macrophages wrapping around these early tumor cells. We see them sending out and retracting processes. And these processes are long-lasting, as I mentioned. And importantly, we almost never observe phagocytosis here. So these interactions appear to be friendly. And obviously, we speculated that these interactions might contribute to the tumor promoting activity. And we wondered what causes these microglia interactions and what are the functional consequences here. And what we speculate here is that these early neoplastic cells might actually hijack a mechanism that is usually employed by healthy, viable neurons to attract microglia. And this is summarized here. So what's been shown in a variety of models over the past years is that highly active neurons with increased calcium levels release ATP into the extracellular space, and the microglia express a specific receptor called P2Y12, which allows them to sense the ATP, and that's the way how they interact with these neurons. So we were speculating that these early neoplastic cells might actually use the same mechanism to attract the microglia. And we have started addressing these questions by using another transgenic setup here, which allows us to visualize calcium levels in these early neoplastic cells based on a GCAMP reporter, as shown here. And as you can see, in comparison to the control cells, these early neoplastic cells have significantly increased calcium levels over the time. So we can show there's more calcium in these cells. We can also show that the microglia respond to these cells that increase their calcium levels. So if you just follow the cell within this white box, you can see that this cell will increase its calcium levels, and the neighboring microglia cell will be directly attracted to this cell. We followed up here, as introduced, these microglia express a specific receptor called P2x12, which allows them to sense ATP. So we have generated a mutant for the receptor and addressed these interactions between microglia and the neoplastic cells. As you can see here in the control and the wild type, we get this increase in microglia interactions as expected. However, upon knockdown of the P2y12 receptor, we don't get any increase in the number of interactions here, meaning the microglia really need the P2y12 receptor. So in summary, we have preliminary evidence here that these pre-neoplastic cells might actually hijack the calcium ATP P2x12 receptor mechanism to stimulate these interactions. We're currently looking at the functional impacts here to prove our hypothesis that interactions will stimulate proliferation. Let me now briefly summarize. So basically what we can show is that these early tumors in the brain, these so-called pre-neoplastic cells that just become abnormal, these cells release SDF1B, which leads to the attraction of macrophages via the CXCR4 receptor. These macrophages infiltrate the brain, and then some of these cells differentiate into microglia-like cells. I've called them brain engrafting macrophages here, and we see an immediate pro-tumoral action of these macrophages here. Finally, let me thank my team in Edinburgh for their hard work on the project, my collaborators on the different projects listed here, and a big thank you to Cancer Research UK for funding our work. And thank you for listening.